acids, the building blocks of proteins, link together in linear chains through peptide bonds to form polypeptides. But forming a simple chain is not enough. The next step in protein synthesis is folding that polypeptide into its specific shape or conformation, and then in some cases combining multiple polypeptides to create a larger functional protein. Now, what really drives this whole process and keeps the protein stable? Well, a variety of interactions between different parts of the molecule, carboxyl groups, amine groups, and the R groups of amino acids play a crucial role. Let's go through them one by one. First, we have disulfide bonds. These are covalent bonds formed between the sulfur atoms of two cysteine residues. Take a look at cysteine. You'll notice a sulfahydryl group in its R chain. When two cysteines come together, a disulfide bond forms through an oxidation reaction that removes two hydrogen atoms. Because it's covalent, this bond provides significant stability to the protein structure. The only way to break it is by reducing it, essentially reversing the process by reintroducing the hydrogen atoms and restoring the sulfahydryl groups. The sulfide bonds can form within the same polypeptide chain when two cysteines that were initially far apart get close due to natural folding, or between separate polypeptide chains, covalently linking them. When they form within the same chain, we call them intermolecular disulfide bonds. When they form between different chains, they are intermolecular disulfide bonds. Now, the other types of interactions we are going to talk about are non-covalent. Individually, they are much weaker than the sulfide bonds, but since there are so many of them, they collectively have a huge impact on protein stability, especially inside cells. Let's start with hydrogen bonds. In order to have an hydrogen bond, we require a donor and an acceptor, two groups in the amino acid chain that might be far apart initially, but are brought together as the polypeptide folds. The donor has to have a hydrogen atom covalently bonded to an electronegative atom, like oxygen or nitrogen. The acceptor, on the other hand, must have an electronegative atom that can attract that hydrogen. A good example, hydroxyl and amine groups make great donors, while carbonyl and sulfahydryl groups are great acceptors. Thanks to the abundance of these functional groups, hydrogen bonds are everywhere in proteins. Even though a single hydrogen bond is much weaker than a covalent disulfide bond, they are crucial for maintaining protein stability. Next up, ionic bonds, also called electrostatic interactions. These happen when the R groups of different amino acids are charged. Some amino acids have side chains that are positively charged, while others are negatively charged. Naturally, opposite charges will attract themselves, while same charges will repel themselves. What's interesting about ionic bonds is that they can act over longer distances compared to many other non-covalent interactions, but they are also highly sensitive to pH changes. If the pH shifts significantly, the charges on the groups involved can change, disrupting the bonds and contributing to protein denaturation. Then we have van der Waals interactions. These are also charge-based, of course, but they are much weaker and more short-lived. They occur between temporary dipoles, that is to say, regions of molecules with nonpolar covalent bonds that, for a brief moment, develop slight positive and negative charges. When two uncharged atoms get really close to each other, their electrons start to interact. The shifting positions of the electrons around the nucleus can create an electric dipole, which in turn induces the formation of an opposite dipole in the other atom. If two molecules with these temporary dipoles get extremely close and are correctly aligned, which is important, they attract each other, but only for a fleeting moment. Finally, we have hydrophobic interactions. These depend on the solubility of the amino acids. The R groups of amino acids can be either polar or nonpolar, meaning they can be hydrophilic, they will love water, or hydrophobic, they will hate water. Hydrophilic amino acids will tend to locate themselves towards the surface of the protein. A protein with mostly hydrophilic amino acids will tend to have a more open structure, allowing maximum interactions with water and other polar molecules. But what about a protein with a lot of hydrophobic amino acids? That one will prefer non-polar environments, like the interior of a membrane. Most polypeptide chains contain both hydrophilic and hydrophobic amino acids, though, so a protein's final shape is a balancing act. You can better visualize it here. We have this polypeptide that is not folded. The green dots are the hydrophobic amino acids, the light blue dots are the hydrophilic amino acids. When the protein folds, as we said, hydrophilic groups tend to stay near the surface, while the hydrophobic groups cluster inside. And these are the bonds involved in keeping the proteins together, stable and functional. Here's a quick recap. Thank you for watching, please like and subscribe for more, and happy studying!